This video explains how to cryopreserve human iPS cells cultured in a 100 mm dish under feeder-free conditions. Take out the culture dish in which the human iPS cells have been cultured from the incubator. Check the cell growth under a microscope, and transport the cells to a safety cabinet. Remove the medium from the dish which the iPS cells have been cultured using an aspirator. Wash the dish with 6 ml of PBS and remove the PBS with an aspirator. Add 1.8 ml of 50% triple select solution and spread on the surface of the dish. Transfer the culture dish into a 37 degrees Celsius, 5% CO2 incubator. After 1 minute, the dish is taken from the incubator and spread the 50% triple select solution throughout the dish surface again and return into the incubator. After reacting for an additional 3 minutes, for a total of 4 minutes, the dish is taken from the incubator and observe the cells under a microscope. At that time, check that the cell-cell adhesion has weakened and that each cell is rounded. Normally, a treatment time of 4 minutes does not decompose the adhesion between the iPS cells and the coated matrix, so it can be observed that iPS cells remain attached to the dish surface. If the cells-to-cells still adhere tightly after 4 minutes, the dish should be returned to the incubator and check the condition every minute until each cell is rounded. After incubation, transfer the culture dish to a safety cabinet and aspirate the 50% triple select solution. Then wash the dish surface with 12 ml of PBS and remove the PBS with an aspirator. At this time, PBS should be added gently, since iPS cells are easily detached. After washing, add 4.5 ml of stem-fit medium. Then, iPS cells are peeled off with a cell scraper. Disperse iPS colonies by 10 times pipetting gently with a micropipette attached to P5000 tip to separate iPS cell colonies to single cells. And transfer them into a new tube. Add 5.5 milliliters of stem-fed medium to the dish, and wash the dish. Washed medium is gotten together into the tube which the iPS cells were previously collected. Sample 10 microliters of cell suspension, mix with an equal volume of 0.4% trypin blue solution, and measure cell density with a cell counter. Transfer 8 million viable cells or more to a 15 milliliters tube and centrifuge at 160 G for 5 minutes. Mark the cell name or label on the cryotube during centrifugation, and prepare for dispensing. After centrifugation, remove the supernatant with an aspirator and tap the cell pellet to break it. Resuspend in a stem cell bunker to a cell density of 1 million cells per milliliter. Dispense 200 microliters of the cell suspension into each cryotubes using a micropipette attached to P1000 tip. The iPS cells should be dispensed as soon as possible, since they will be damaged gradually after being suspended in the stem cell bunker. After dispensing, place the cryotube in the cell freezing container, and then carry it into a -80 degrees Celsius freezer and freeze slowly. After freezing at least 3 hours in the freezer, transfer into liquid nitrogen preserve tank at the same day or the next day and store.